So thank you for your patiently waiting. We've had some technical issues with the mic. I don't know if this is working. It's working? Okay, perfect. Thank you so much for coming, taking your time, fighting the traffic. I just wanted to introduce myself. I am Vidyodhya Sundaram. I am the VP of BizDev at BioChain. I have been at BioChain for more than 6 years. It has been a wonderful journey at BioChain, and we are excited to launch the first inaugural spatial biology symposium because BioChain now has been providing spatial biology services. So this is something that we wanted to talk about, introduce our services, and also our partners. So I would actually personally like to thank all our partners who are here from 10x. We provide 10x Visium, NanoString. I see the entire crew from NanoString here. Thank you. Shout out for people coming from Southern California, Debbie and Cecilia, and Praveer. I know it was just local, but still thank you so much for coming. And we have the team from Enable. Thank you, Gurpreet, for coming in, and our team from Stormix over here. We have a large presence. We are introducing, we are onboarding Stormix at BioChain. So I would truly appreciate all of us, all of you coming here. And of course, the others who've signed up to come in person. I hope this is going to be a great learning. You'll learn a lot of different platforms during this symposium. And also all the people who've joined in on Zoom for the virtual people. I appreciate people joining in, signing in. So I would like to start off with an announcement today that we are excited to officially announce a partnership with Enable Medicine, where BioChain and Enable Medicine now have partnered. There are a couple of things that we are doing with Enable Medicine. The first thing is since we do a few spatial biology platforms such as 10x NanoString, and Enable Medicine does Akoya. So this would be a great opportunity for customers to come in and say, hey, if you're interested in having multimodal systems, spatial systems, this would be a great partnership. So they come to either one of us and say, hey, if you are doing Akoya, then if you're interested in Visium or NanoString, we could partner with that. The second part, which is really exciting, is their spatial bioanalysis. They have an incredible, very powerful bioanalysis platform, and currently they are providing us with 10x Visium spatial analysis platform. So I think this is a fantastic relationship, and we hope to build more pipelines. So we will start adding other pipelines that we are, as we start to onboard new spatial technologies on BioChain. So I appreciate this. It's been going on for a while, and finally, we'd like to announce this over here. And coming to the logistics of today's symposium, we will have the first two presentations in the morning. Then there will be a lunch break and a little bit of time for everybody to socialize. And then the next three discussions or presentations. In between, there will be Q&A sessions. We will try our best to answer both from the virtual as well as the in-person. And if there are some technical errors, just bear with us. And if there are questions that the virtual people have, if we are unable to answer them, we will reach out to you directly and help you answering those questions. In person, if we don't get to it, the entire team is here, BioChain's entire team is here, and every one of our partners teams are here. So please reach out to them as as we throughout the day, and they will be happy to assist you. So to start, the first presentation is going to be from Enable Medicine. I would like to first introduce our presenter, Dr. Gurpreet Kaur, who is a Business Development Director at Enable Medicine. Gurpreet Kaur covers the West Coast and Central US. She's completed her PhD in cell and molecular biology at the University of Wisconsin-Madison, where she studied organelle biology using live cell imaging and also taught at the Vision Research Microscopy Core Facility. The title of her presentation is From Images to Insights, Deep Spatial Profiling Reveals Disease and Treatment Biomarkers. Thank you.
Right. Hello, everyone, and thank you for being here. My name is Gulpreet, and thank you, Vidya, for the really nice introduction. And I'm from Enable Medicine. I'm the Business Development Director for the West Coast and Central, and we are really excited to partner with BioChain. They're one of our many, many partners, but this is a personal one too, and we really love working with them. So I'm going to talk a little bit about who we are, what we do, and then I'll show you a couple of case studies of some of the work that we've completed. Hopefully that gives you an idea of the kinds of work we can do for you. So primarily at Enable Medicine, we are a team of systems immunologists, engineers, bioinformaticians. And one of our goals is to improve outcomes for clinical trials and enable biological research. And we want to do that using our scientific expertise in systems biology and in computer vision. So how can we improve outcomes for clinical trials? One of the ways is by using pre-selection biomarkers. I want to show you some data showing phase transitions for clinical trials from phase 2 to phase 3 over a 10-year period. So when these studies used pre-selection biomarkers, there was a 20% increase in their chances of going from phase 2 to phase 3. This data is actually from this report called Clinical Development Success Rates. And this report also actually came up with a metric for likelihood of approval of a therapy or a drug. And they said that programs that employed these patient pre-selection biomarkers actually had a 2-fold higher likelihood of approval that was driven primarily by their phase 2 success rate. So having said that, what biomarkers can we use? This study, over the last 10 years uses all kinds of biomarkers, physiological biomarkers. But now we're moving slowly into molecular single cell spatial biomarkers, right? So what can we do to actually help clinical trials? So the first thing we're doing is discover novel biomarkers using single cell and spatial techniques. Doing so, we can actually evaluate the mechanism of action by actually testing different proteins and transcripts at the same time and putting them together. We're able to come up with outcome prediction. So we have our bioinformatics pipeline that can actually predict treatment outcomes and predict biomarker outcomes and see how relevant a biomarker would be to a given therapeutic. And finally, we're able to stratify patients based on these. You can figure out what were the differences between responders versus non-responders, what changed between a disease score of 0, 1, 2, 3, all of those really nice questions. So we're doing this by leveraging the power of spatial biology. On patient tissues, where we image and annotate H&E images, as is quite common in most pathological laboratories. And then we're able to correlate this data with hyperplex immunofluorescence staining. We use a discovery panel of like 51 biomarkers, and we can add a couple of custom markers also that actually tests various checkpoint markers, epithelial and tumor markers, different antigen presentation markers. And we're able to come up with different cellular niches and neighborhoods using proteins on tissue. And at the same time, using our partners like BioChain, we're able to do a lot of different transcriptomic data generation. And finally, using our pipeline, we're able to integrate all of this. So I'm going to talk to you a little bit about one of our methods that we've recently developed to integrate different types of data. It's called EM Object, and this manuscript was actually just released on bioRxiv last week. EM Object is essentially a data abstraction for spatial omics. It's agnostic of different data types, so you can analyze Visium, maybe Codex, geomics data sets really, really easily. And I'll just walk through exactly what it does and give you a higher level introduction to this. So basically, if you had any of these data layers, so you could have high-plex imaging, low-plex imaging, or like bulk gene expression, it uses one of the layers as the core of the data matrix. And then all the other layers are put in as attributes. So if you had a Visium layer and a Codex layer, one would be considered the core and the other one would be considered an attribute. And then there's a special attribute for like the spatial omics, the spatial data, the spatial coordinates. And then there's a transformation and alignment, which means you could align the data sets into this core organization before analyzing it or after analyzing it. So you can switch them either way. And finally, along with the EM object, there's an EM experiment, which means you can do multiple cohorts of multiple EM objects and put together your full data set. So showing you what a complete workflow would look like. So if we were generating our hyperplex imaging data using our Akoya panel, we would have these 51-plex biomarkers and custom biomarkers for whatever targets you have. We can include transcriptomic data from BioChain. And then it goes into our own analysis pipeline where we do segmentation, we have our pipeline for quality control, and then we do unsupervised clustering to actually figure out what these different cell types are, and we phenotype them on our own. And finally, we figure out spatial neighborhoods. So we figure out cellular niches in which these cells are acting and forming active niches that are contributing to the biology. And finally, we have our Explorer. So this is a really cool system, how we deliver our data to all our customers. It's an interactive portal. We're looking at multi-omics data at this point. Obviously, we'll come up with 2-3 main stories that make sense, a couple novel targets that make sense for your data. But there is so much data to parse through. There's always going to be more in your data set. So the Explorer actually is an interactive portal that comes up with graphs. So you yourself can go in and stratify. All right, I want to look at the difference between responders and non-responders, but only in this specific condition. And it'll pull up these graphs for you. So even years later, if you wanted to go back to your discovery experiment and see if there was any relevance, you can actually do that. And we have built multiples of papers based off of just one key experiment, just because there's so much data over there. And the Explorer makes it really easy to go through all of that. Right. So doing all that, we are answering the core biological questions. So these include what are novel therapeutic targets for disease indications, what biomarkers can we identify that predict response to therapy, what biological mechanisms drive these pathogenesis or therapeutic responses, and finally, what is the optimal therapy for a given patient. To illustrate this, I have 2 case studies here that I'd like to walk through. The first one is actually a phase 1 clinical trial for a large pharma company. So we completed this for last year for a pretty big pharma company, and they had minor adverse effects in their phase 1 clinical trial. This was an immunotherapy trial, and they got some like skin inflammation, which is quite common for immunotherapy trials. But they were quite concerned about this, about how to get this approved and how to move forward with their drug, and essentially wanted us to sort of rescue it. We went in to understand what was the mechanism of toxicity, what that was driving this, in this drug or therapeutic. For the phase 1 trial, we obtained affected and unaffected patient samples, and we imaged them with our discovery panel of 51 biomarkers, and we actually included 2 custom biomarkers that were specific to this pharma company's target. After doing that, we were able to do spatial analysis where we looked at different cell types, cellular interactions, and cellular neighborhoods that were present in this, in the entire data set. We were able to identify a rare cell subtype that was associated with the mechanism of toxicity. Now, I can't reveal a lot of these markers, but I'm going to walk you through some of the data that might illustrate exactly what the answer should look like. So I'm going to show you these Warnock plots. These are pretty common for us to produce. So you start by imaging, so you have all these like protein markers, right, that you're looking at in immunofluorescence. But this is actually output of that. Now now we've gone in and identified each and individual cell. Here's a list of cells that are present. These are skin samples. So you can see the epidermis at the top, dermis at the bottom, and you can start to like really just see where all the different cells are and what they're interacting with. And these are the two target cells that we were primarily interested in looking at. There's a target positive macrophage and there's a cell subset Y. Finally, we can start quantifying these. So these are Warnoy plots. So everything that's enriched in the affected samples is on the left here, and everything that's enriched in the unaffected samples is on the right here. And this red line is, showing significance. Anything that's above this red line is below 0.45. So here you see that rare macrophage subset that we were talking about, and then CD4 positive T cells, as was expected because we saw some inflammation. And then we have some graphs that show the actual distribution of these also. So just want to walk you through the steps that happened here. So the company had H&E results. They knew that affected patients had T cell infiltration based on just H&E. We completed the hyperplex imaging, and just based off of that, we were able to identify what types of T cells. So we found out that these were CD4 positive T cells. And then we were able to identify this rare macrophage cell type that was specific to the target. And we were able to generate the hypothesis for the mechanism of toxicity. So the idea is that the skin samples in affected patients express this rare macrophage phenotype that's expressing this target protein, and that's why they're getting these adverse effects. So going from there, this company can now either use that as a potential biomarker, or they can simply screen against this biomarker. So now they can just use like a panel of 3 proteins if they want, test, all right, this marker, this marker, this marker. If a person has that, they don't include that in their next trial. So you kind of push your drug forward, therapeutic forward, and able to stratify the patient that it's going to work. So just concluding this with what we're able to do. So we just did discovery analysis, discovery panel and spatial analysis. And we were intersecting with this phase 1 environment of multiple different countries and continents and different sites. And we were able to manage all of that and actually figure out the differences between the cellular phenotypes of affected and unaffected patients and indicate the information for this company. And it's really interesting because it was hard to pinpoint causality because they didn't have pre-treatment, they didn't have pre-results, right? So you can't say, oh, before and after. You can't do that in humans. So, but we're able to give them really highly indicative results, and they're able to use that forward. And now they're able to build their next set of tests. So that was the first story I want to say. And then I actually want to highlight this second story. It's quite interesting because this was done in collaboration with Arutha Kulasinghe from University of Queensland. And this paper was recently published, and actually our friends from NanoString also are employing, are, I guess, part of this paper, right? So I'm going to walk you through really quickly just to illustrate how powerful multi-omic data can be. Here we were able to obtain samples, immunotherapy samples for NSCLC. So these are advanced stage NSCLC patients that underwent immune checkpoint inhibitor treatment. And then we had non-responders versus responders. And Arutha's lab and us, and actually in collaboration with NanoString, Vicoya, did multiplex IHC, codex, and geomics on this data set. And the question was to really identify what cell types of interactions separate the responders from the non-responders, and if we can identify any key biomarkers from all of these techniques. Here's some representative images for what a non-responder versus responder looks like. This was published. But I want to talk to you about the process going forward for the proteomic analysis. So when they just looked at the cellular profile, we didn't really find any differences in this cellular profile. So no real cell type differences between responders versus non-responders. And so the next step was to look at what are these cells interacting with. And when we started doing that, we actually found that there was this population of CD68 positive macrophages that was interacting with PD1 positive FOXP3 positive cells. So these are Treg cells that are positive for PD1. So there was some interaction going on over there, and we were able to explore how immune checkpoint inhibitors are actually targeting, are actually interacting with these CD8 positive cells, Tregs, FOXP3 positive cells, and macrophages. So the point I want to make with this is that these spatial relationships are a really important metric to assess the cellular profile of patients. We've been talking about single cell data for a while. We've been looking at cellular types for a while, but this really illustrates that we need to go beyond that and look at what these different cells are interacting with. All right. The next thing I wanted to show was that when they did the geomics on this, so when they went in and used the transcriptomic analyses, they were able to come up with a couple of different transcripts that were upregulated, downregulated, and also correlate those to the proteins that were downregulated. So actually increases confidence in the entire data set when you're seeing multiple assays give you the same. So I'll walk you quickly through some of these results, and then I want to talk about the actual correlation here. So in the immune checkpoint inhibitor responders, they saw that the CD44 protein was downregulated, and SPP1 mRNA, which is actually a ligand for CD44, was increased, right? It was quite interesting because SPP1 is thought to be secreted by some myeloid cells, but they didn't really see an increase in the myeloid cell type as such. But SPP1 is implicated as an immune suppressor, right? And CD44 is a hallmark for cancer cell stemness, so it promotes cancer survival. So finally, they were able to come up with this hypothesis that in non-responders, it's possible that the soluble osteopontin is interacting with the CD44 receptor. It's inactivating CD8 T cell cytotoxicity, acting as an immune suppressor, right? So they're able to build that, but I want to actually also point to this graph over here. So this is codex-based mean fluorescence intensity, so expression levels of CD44 on the y-axis and digital spatial profiler counts on the x-axis. So we're correlating transcriptomic and proteomic data sets right now. That's a pretty nice linear relationship. The R is 0.61. I think like for different assays, that's pretty strong to start with. So in conclusion, I want to say that this is actually a really small cohort that we started with. And we have next analyses going on right now where we're actually truly integrating the data sets and seeing if there are cellular niches that we can go in and look at. But even with just this, we were able to come up with some novel biomarkers that were actually responsive for these immunotherapy responders. We're able to differentiate just the cellular phenotypes with responders and non-responders and get just a lot of novel information from even a small assay over here. All right, concluding that, I just want to say that, enable medicine, we're interrogating different patient tissues, we're exploring spatial profiles with single cell resolution, and hoping to enhance our understanding of disease and improve treatment outcomes. And we have worked with a lot of researchers around the globe. We have lots of academic partners and lots of industry partners, and we're excited to work with a lot of new ones too. All right, thank you.
Questions? Thank you so much. So we are going to take like a quick short, 2-minute break or 5-minute break for the next speaker to come in. Thank you very much.
I see the live stream wasn't getting it. Welcome to the people on the live stream. I hope the pictures did a good enough job so far. So as I mentioned, we can see really interesting gene expression differences in different parts of the kidney. More recently, of course, profiling COVID-19 samples has been of great interest. So these are from lung tissues, and we can go in deep into different parts of lung tissue. We can pick infected versus uninfected cells. We can even use viral gene expression to guide us in what cells we're picking, as shown here. So we're segmenting on viral gene expression itself. Studying more basic biology, we can go in and make very detailed atlases. So this is a whole transcriptome spatial atlas of mouse development across a number of critical days. And we can go in, look at all sorts of different tissues, and once again, get whole transcriptome expression here. We have on our website already full data sets for 5 human tissues, 2 mouse tissues. You can go in, you can download them, look at them, and use them to guide your own thinking. A couple of case studies I want to mention. This is work from Australia, Rachel Kolday's lab, looking at bone marrow and looking at maturation of different immune cell types. She said this will allow, even in patient cohorts, identification of new biomarkers and new mechanisms. David Rim at Yale was looking at checkpoint inhibitors. Of course, Gulpreet mentioned them. They're a very, very hot topic of research. And looking at non-small cell lung cancer, finding really interesting differences in different compartments. And then work from Goran Jansson's lab in Sweden, looking at B and T cell interplay in a way that's really hard to do before because you don't have that high level, full transcriptome level information. So genomics has been really embraced by the research community. We have, I think we're at numbers 170 plus now, just keeps going up. So we're very pleased with its popularity. Now we'll talk a little bit about how it works and how you actually put it to use. We start with, in very brief, we start with samples. We expose them to our probes. We put them on the instrument, select our regions of interest, we collect the signal, and then we count it on a couple of different options. So I'm going to go into each of these in a bit more detail. First step is, of course, to prepare the tissue. And we can work with any type of tissue. That does include FFPE tissue from a biobank, frozen tissue, biopsies, tissue microarrays. Really anything you can put on a slide, we can work with. Take the tissue, put it through standard workflows, deparaffinization, and so on. And then we add 2 sets of reagents. One is the imaging reagents. These can be antibodies or it can be RNA scope probes. And then we have our profiling reagents, and these are what will actually generate the signal. Once again, antibodies for protein targets, oligo probes for RNA targets, and these can be done simultaneously as well. These are the profiling reagents in a bit more detail. For protein, we have an antibody that binds our target protein. There's a photocleavable linker, and then an oligonucleotide barcode. For RNA, once again, we have our target transcript here, complementary sequence, which will go bind to the target, another photocleavable linker, and then an oligonucleotide barcode, similar to the protein. Proteins, we can look at up to 150 targets at once, and these can be based on our panels, or these can be custom. For RNA, we can have up to the whole transcriptome. For humans, we're looking at 18,000 targets. For mouse, 21,000. And these can also be customized. So they can be customized on top of the existing panel, or you can make fully custom panels. Once the samples are prepped, we take them on the instrument. This is where those imaging reagents that I'd mentioned come in. So this is the colorectal cancer sample. You can see here we have it stained with a DNA stain. This is cyto13 and pan-CK and CD45. And this is standard IF type antibodies. And I hope you can appreciate how good the imaging quality is. Even though it's not an imaging instrument per se, it is in fact a really good microscope on top of everything else. And we can have a whole tissue like this, and then we can zoom all the way in and see in quite a bit of detail. So here we go in, we select our regions of interest, we set up our different segments, we can draw any kind of custom shapes we want, and then we can do segmentation based on these imaging markers, or we can use an overlay like an H&E overlay on top to guide our regions of interest. So as I mentioned, we can draw any shape. Once at a training, I had someone draw a dinosaur on top of their tissue. We can do segmentation based on markers. We can pick out specific cell types based on different markers they might be expressing. We can do these contour shapes. So we can define an initial shape and then pick regions going out from that. Or if you want to be completely unbiased, you can just put down a grid that collects everything from a certain area in small bins with nothing missed. So all of the cells have a collection happening from them. And we collect signals. So as you remember, our probes, whether they be for RNA or protein, have this photocleavable linker and then an oligonucleotide. And inside the instrument, we have this micro mirror device, which has about 1 million small mirrors, each about 1 micron square in size, that can be individually controlled. So we can shine UV very, very specifically on just the regions that you define. Even if you have 2 cell types that are right next to each other, we can still reliably and accurately pick signal from them separately. Get really, really clean signal. We'll go in there, we'll cleave off these oligos in exactly the region that we have defined. And then these oligos get picked up and deposited into a 96-well plate. The oligos themselves are barcodes that are compatible with the standard Illumina type sequencing workflow. So we add the primers, make a library, clean it up, send it off for sequencing. Once the sequences are ready, we bring them back on to the instrument where the counts are integrated with the imaging data, the run that was done on the instrument itself, and then we can go into analysis. That can be done on instrument, off instrument, from home, on a phone. It's not great on a phone, but you can do it. And you can export the data into RStudio, into any other analytic pipeline you want as well. And what's built in, standard things, you can make PCAs and UMaps, heat maps based on many different types of clustering, differential expression. Something like this, we did it internally, we really liked. You can go into a brain and select very specific regions and then do a color coding on top of it to show different levels of expression, just at a glance. And the fun part about doing an experiment like this, here we're showing 1 gene. Remember, the run was the whole transcriptome. So you can look at any gene that is expressed in this tissue and be able to make a map like this from 1 run. So something that even a few years ago would have taken a postdoc 6 years to do, now you can get done in 2 weeks. The performance is really tight when you do runs even on different dates. Do one and then you do another one a couple of weeks later. The counts are right on top of each other. The reproducibility is great. Here, the numbers here, the sizes are diameters of the regions of interest. So the larger you get, the more signal you're collecting, and your reproducibility gets a bit tighter too. But even when it's quite small, and 50 microns where you're collecting maybe a few dozen cells for the RNA assay, this is a whole transcriptome assay, it still looks great. It also correlates well with orthogonal sources, both human and mouse here, comparing to RNA-seq and comparing to single molecule in situ as well. This was RNA-scope. And then the readout, it can be done on both, on either next-gen sequencing, specifically for the whole transcriptome assays, that is the outcome. Or if you're doing one of the smaller assays, you can do the readout on our nCounter platform. So it's a bit faster than through NGS, depending on your use case. I'll talk a little bit more about the panel. As I mentioned, we have the whole transcriptome assay. It's not 3' biased. It's based on designed probes that go to the targets. And for human and mouse, we do cover every target. We take out a few very high expressors, beta-actin type stuff, just so they don't swamp the signal. But if you really want to look at them, we can add them back in. As I mentioned, you can look at the morphology with IF or with RNA-scope. Works with any type of tissue. And I want to emphasize, why does this work so well with FFPE? It's because we're not doing anything enzymatic. It doesn't matter if the RNA is fragmented. We just need these small regions where our probe can bind. It's less than 50 nucleotides that it needs to bind, and so it works great with FFP. You can, of course, detect non-protein coding genes, any transgene that you're expressing, anything else that might be in there, infection. You just tell us what you want to look at, and we'll design a probe for you. This is where the customization comes in. You can have up to 400 custom targets, even on top of the whole transcriptome assay. For the mouse one, already a lot of common transgenes are included in there, but of course you can add things on top. The Cancer Transcriptome Atlas is sort of a smaller version of our whole transcriptome atlas. This is 18,000 targets. This is 1,800 targets, focused on cancer, as the name suggests. Protein assay, we can look at, as I mentioned, up to 150 targets if you're doing an NGS readout, or 96 targets if you're doing an N-counter readout. And it comes in these 10 target modules that you can mix and match for immunology, IO, and neuroscience projects. And the antibodies, we already tested them to make sure they all play well together. But you can add, of course, antibodies on top of that. This is just a quick overview of some of the panels that we come with, and of course, all the targets and for all the panels are fully available. You can see them on our website. You can ask one of us, and we'll send them to you. Nothing proprietary about that. I won't spend too long on these slides, but you can see that we have everything, and chances are your favorite target is probably in there already. Neuro1. Then there are mouse versions of all of these too. If you're ordering an antibody, or even this is similar to how we do it internally, we have to, of course, conjugate the antibody with that oligo. So we do the conjugation, then we test it. We test it internally. We test it together with other things. So we make sure it works properly. And you can do these custom antibodies. We have a partnership with Abchem. They can do it for you, or you buy your own antibody, then you send it to us and we'll barcode it for you. So you have a couple of different options. A little bit more about that NGS readout. As I mentioned, when you're doing one of our assays that has an NGS readout, we'll make a library and send it for sequencing. So each region that was illuminated on your sample, that is one well. But when we collect them and we make the library, each well gets tagged with different indices so that you can combine everything together for one big sequencing run. So the indices here, the I5, I7, they all go in separately to each well. And we provide a plate that has all the primers, so you don't have to pipette out 100 different things. You just use a multi-channel and everything gets mixed. And then after the sequencing is done, it goes into a pipeline that we provide, aligns, cleans them up, trims the ends, and then that goes into our analysis pipeline. We do QC, background normalization, and then into the analysis. And depending on the project you're doing, depending on the kind of assay, you can have different levels of readout, different numbers of reads that you would need. So a whole transcriptome run, usually we end up seeing 1 billion plus reads. So NextSeq or NovaSeq work just fine. Cancer Transcriptome Atlas, you can do it on a NextSeq. The same thing with protein, really depends on how many targets you're looking at and how much area you're collecting on your tissue. This is just a glimpse of what the analysis looks like. Once the sequencing is done, counts are loaded on here. You look at your tissue, you have the nice images, and then you have your regions of interest highlighted there. Go in and look at them. You have all your targets listed here, and then you have your analysis output on the right side. And you can really combine a lot of different things. You can put your overlays from other sources of imaging on top. You can include reference matrices, for example, from RNA-seq, clinical annotations for different samples, and then everything gets pulled into the analysis. And it really enables collaboration in a lot of ways. Person who's operating the instrument doesn't necessarily have to be doing the region of interest selection. Pathologists could do that remotely. They can do it from their office. They don't need to show up on site. Once the data is analyzed, anyone can log in remotely to look at it, run the analysis, look at the results. So you don't really need to have anyone on site to be able to do this. If you're running it on Vidya's instrument, you don't have to personally go to New York. Although the food there is pretty good, and you can have it done remotely just fine. In summary, with GeoMx DSP, you can do really great spatial work, lots of targets, both RNA and protein, on any type of tissue. And we're working with a lot of partners. Of course, BioChain is one of our CRO partners. With readout, we work with Illumina, working with Azenta, what Gene was used to be, for library prep. We have a partnership with Visiopharm for that ROI selection, enabling it to be AI-driven. The sample prep can be fully automated on a Leica Bond. As I mentioned, we're working with Abcam for antibodies and RNAscope for morphology visualization. So once again, it's a really, really complete solution for spatial biology. But there's one thing it does not do very well, and that's single cell. And that's where I'm going to lead to next. With GeoMx, we work with groups of cells at a time. These can be as little as 5 to 10 cells for certain assays, into a few hundred, even a few thousand on the upper end. But usually we're working with groups of cells. And with our morphology markers, you can group them very nicely, very cleanly, and of course, look at the whole transcriptome. But sometimes you really want to look at one cell, or even inside a cell. And that's where CosMx comes in. With the CosMx Spatial Molecular Imager, it was just recently launched, you can look at individual transcripts in single cells and inside single cells. We're looking at single molecule visualization at a 50 nanometer XY resolution. And right now we can look at 1,000 RNAs at a time. That number is going to go up, and I'll talk about that in a second. CosMx, we can look at 1,000 RNAs, single molecule. We can, once again, as with GeoMx, work with any sample type. We can look at RNA and protein, and we control, how many samples you want to run. It's really up to you. You can design your projects in different ways. And it's really high resolution, going inside cells with 3D data collection. Sample prep, once again, straightforward, the way it is for GeoMx. Then you load the slides onto the instrument, and the readout happens on the instrument. So unlike GeoMx, where you have to read out your counts on either an N counter or with NGS, with the CosMx, everything is done on the instrument itself, and you go straight into data analysis. Now, the data generated by the CosMx is very large, and so our solution for that is to send the data directly to the cloud with our Atomic Spatial Informatics portal, and it's stored there, it's shared there, it's analyzed there. So you don't have to worry about where you're going to store these terabytes and terabytes of data. So you can go on the CosMx looking at this whole tissue. There's a 15 by 20 region on the slide that you can place your tissues. You can have a pretty good chunk of tissue, or you can have fairly large TMAs. And just sort of scroll in with your mouse wheel, get into a single cell, and get inside the single cell. So on a single slide, and certainly in a single run, you can be well over 1 million cells that you're collecting data from simultaneously. That's 1,000 right now. With protein, we're looking at 64 targets, and then there's morphology markers on top of that. It's not quite single molecule the same way it is for RNA. Proteins are usually a lot more abundant. They're tightly packed, but you can still get inside a cell and ask regional questions inside cells. I'm not going to spend too much time on this, but we can really look at a lot of different things. Cell atlasing, of course, cell typing, but we can ask questions, not just what type of cell it is, but what is it doing right now? What state of its cell cycle is it in? What type of signaling is it doing at this moment? Really ask detailed questions. You can ask questions about the microenvironment. What is a cell that's next to a certain neighbor doing that's different from a same cell type that's next to a different neighbor? Disease states, of course, we can look at questions about infection, any kind of pathology. And because we're looking at single cells and we know where they are right next to each other, we can say if one cell is expressing a ligand and the other cell is expressing a receptor that's next to it, there's probably a ligand receptor interaction occurring, and we can measure that, quantify it, and really look at cell-cell interaction of all sorts. I'm happy to discuss these in any more. The chemistry is somewhat similar to genomics, but also a little bit different. We have our target molecule, RNA or protein. Then one probe comes and binds. That's an oligo or ISH probe, as it says here, for RNA targets, and an antibody for the proteins. And there are these other binding sites. These are just sequences where a second probe that actually carries the fluorescence signal comes and binds. This first probe is bound during slide prep. The second probe happens on the instrument. Same thing with the antibody. There's a linker to an oligo, which has a landing site for the receptors. So the imaging happens on the instrument, and even though it's just 4-color imaging, by doing it in cycles, sequential barcoding, we can get to 1,000 targets just with these colors, just 4 standard colors. Works with all cell types just fine. FFP, of course, microarrays, section organoids, fresh frozen, mouse brain example here. Again, we have assays, talks about a little birdie here, where we have a human IO focus panel that's 100 targets at a time, a human universal panel that's 1,000 targets, a mouse neuroscience panel that's also 1,000 targets in RNA. And apologize if you have difficulty reading this. This is a human immuno-oncology focus panel with 64 protein targets. We can customize our panels also. Once again, we can add up to 50 targets to the 1,000-plex RNA panel, 10 to the 100-plex panel. For the protein panel, we don't have it right now, but later this year, we'll be able to do 8 custom targets for the protein panel as well. And you can also create a fully custom RNA panel of up to 350 targets. Genomics and cosmics work very well together. They can feed into each other if you're doing a project, trading off the whole transcriptome with a single resolution. For example, you can use genomics to create a predictive immune profile on a type of tissue, and then a more quantitative profile for the same tissue, and combine these data sets for biomarker discovery. You can look at the whole transcriptome expression in infected versus uninfected on the genomics, and then use that to look at individual cells in more detail and pick up on specific genes. You can read more about the technology in our Nature Biotech paper that came out last year. And you can go on our website. Just as with genomics, you can find data sets that we've made publicly available. For cosmics, we have a human lung cancer data set and a human liver data set. And what we have coming next is more assays, 1,000-plex for mouse universal, 1,000-plex for human neuro, a 64-plex mouse neuro panel, and as I mentioned, 8 custom protein targets. And we're really excited next year to make a big jump in how many targets we can look at on the same instruments. We'll be able to look at 6,000 RNA targets and 120 protein targets. With that, I'd like to thank you for coming. I'll point out our team here. Debbie Higbee, sitting right there, is our consumables representative. Brian Witt and Alistair Ruggiero are not here, but they're also part of your team, and I'm, of course, Pradesh Sharma. Thank you very much.
Okay, welcome back, everybody. I hope we had a good time talking, mingling, and having a lunch. Thank you again if somebody new has come in. So we will start off the next session with 10x Genomics, who we are a service partner for 10x Genomics, 10x Visium specifically, and also, as I said previously, for NanoStream. So again, we are that has multiple spatial technologies that we can provide. So to give you a little bit more insight on, this is Samantha Sheldon, who will be presenting 10x Visium technology, earned her PhD in molecular biology from the University of Texas at Austin, studying RNA modifications using a multiomics approach, employing next generation sequencing technologies. She's held multiple roles at 10x, including the technical support and product marketing groups. In her current role as a science and technology advisor, she helps researchers at 10x design their single cell and spatial experiments. The title of her presentation is Envision New Dimensions in Spatial Biology with Visium CytAssist and Xenium in situ platforms. So please welcome Dr. Samantha Sheldon.
So starting with our single cell platform, which is a microfluidics based barcoding solution, which allows you to barcode the transcriptomes of single cells. And so in this plot here, in this plot, what you're seeing is each dot represents a single cell, and these dots are then clustered according to their transcriptomic similarities and differences. And so we don't just stop at transcriptomics with our single cell platform. So we also have a number of other modalities that can be captured. So you can study the epigenome, the cell surface proteome, the immune repertoire, and many other modalities as well using our single cell assays. Our Visium platform uses an analogous technology to barcode spatial gene expression while also maintaining the morphology of the tissue. So you can better understand the gene expression within your tissue while also being able to observe the differences, which areas of different tissue express different genes. And so in this image here, you can see that we have an output from our Visium technology, which allows you to retain this tissue morphology using an H&E image, but also be able to look at the differences in gene expression, and the spatial location of that gene expression as well. So here you're seeing each of the spots is being colored based on, again, the similarities and differences within of the gene expression within that. And finally, we have our Xenium in situ platform. So this is a more targeted readout of hundreds of RNA targets with incredibly high subcellular resolution. This is also a spatial transcriptomics platform, which allows us to look at the spatial location of these RNA targets with single cell and even subcellular resolution. And so today I'll be focusing on our Visium Spatial and Xenium in situ product platforms and the recent updates that we've made to those. Okay, starting with our Visium Spatial platform, which allows you to take something like this, a whole mouse embryo, and be able to look at the different gene expression patterns within that sample, and then map it back to the spatial context. As I'm sure everyone here is very familiar with, spatial analysis is really nothing new. People have been analyzing the histology of tissues since the invention of the microscope. But really, the more recent advancements that have been made in spatial analysis, such as Bayesian scaling, histochemistry, and immunofluorescence labeling, have allowed us to better understand the pathology of these tissues, identify therapies for disease, and identify new diagnostic biomarkers. However, the more traditional technologies that have been used do have some limitations. So sensitivity is a limitation in many of these traditional assays. Your target, whether or not you can detect it, will depend significantly on the quality of the antibody that you're using, if you're using an antibody-based assay, as well as the abundance of the target information. They are often very qualitative, and so it's something where a very experienced user might make one call, whereas a more naive user may make a different call. And so this can lead to mischaracterization and just challenges with analysis of tissue types. Limited multiplexing and limited throughput is also a pretty significant challenge. So, many of these assays have limited abilities to target multiple transcripts. So, or multiple epitopes. So you might get in the Chinese if you're lucky, being able to look at different RNA or different antibody transcripts or antibody signal. And so being able to, this really limits the amount of data and information that you get from your experiments. And finally, the precision of these techniques can vary. So a number of antibodies may have cross-reactivity with different targets, may have false positives in the data, things like that. And so to address some of the limitations posed by these more traditional assays, at 10x, we are really committed to developing our spatial portfolio to address some of those challenges. So starting with our Visium assay. So our Visium assay uses a next generation sequencing readout to detect whole transcriptome gene expression information from fresh frozen FFPE and 6 frozen tissues. We have a couple of different techniques of capturing our gene expression information that I'll talk about in a few slides. But they essentially all use the same tissue slide architecture to analyze the gene expression data. So let's look into that a bit further. First I want to start by demonstrating how Visium can be used to better understand things in the tissue context, such as the tumor microenvironment. So in this particular paper, which is a really beautiful paper, by the way, and it's way more deep than I could even go into in this time allotment. I highly recommend reading it, but I'm just going to cover a few of the findings that we found. So in this paper, the Mayer et al. Were interested in studying these tertiary lymphoid structures within these clear cell renal cell carcinomas. So if you're not familiar, tertiary lymphoid structures, or TLSs, are essentially they're like these structures within tumors that consist of organized aggregates of lymphoid cells that are formed by a network of cytoblasts. And so they essentially produce all these adaptive immune cells within a given tumor. And they're very positive, or they indicate a positive prognostic outcome for these clinical patients. And they also indicate that they are likely to have a positive outcome, particularly with immunotherapy. But that being said, the molecular composition of these structures within the tumors is not fully understood, and the mechanism by which they operate is also not fully understood. And so these researchers wanted to use the Visium assay to better understand how these TLSs function within the tumor microenvironment. So to that end, they took our Visium fed for FFPE and Visium fresh frozen assays, and they used them to assay 24 of these CC-RCC primary tumors. And they were able to do a couple of things using the Visium assay. So the first was that they were able to identify using the Visium gene expression data. They were able to identify a specific gene signature that they could localize to the TLS. So what they were able to do is essentially take, I just use my mouse. They were had a pathologist mark the region where the TLS was present within the tumor. And then within the Visium data, they were able to say, okay, perform differential gene expression on this region of the tumor compared to the rest of the tumor. And so doing that, they were able to pretty easily identify this 29 gene signature panel, which they were able to associate with the TLS signature. And so in this data set here, you can see that this is classified as TLS imprint, and we have a really nice hotspot surrounding the area where the pathologists have indicated the presence of the TLS. When they looked into this data further, they noticed that, of course, there was a number of immune cells, or immune markers present within the TLS. And particularly, they saw a very high number of B cell and particularly mature B cell signatures within the TLS. And so they wanted to examine this a bit further. And so here you can see the presence of these mature B cell markers. So markers are for plasmablasts here, as well as for plasma cells here. Whereas when you look at more naive B cell markers over here, you can see that those are not localized to the TLS in the same way. So the TLS really has a high specificity for these mature B cell markers. And so, like I said, there's a lot more even to this paper than I can go into. But one of the things that they found after looking into these TLS positive tumors using the Visium assay was that these TLS sites are sites for B cell maturation, selection, and clonal amplification of B cells. And that furthermore, they observed that they saw plasma cells essentially traveling along these fibroblastic tracks. So from the TLS, they observed B cells traveling and dispersing through the rest of the tumor. And you can see that here in the plasma cell data over here, where there is a high concentration of plasma cells within the TLS, but also there's pretty significant dispersal throughout the tumor as well. And so this likely contributes to this positive prognostic outcome because these TLS structures allow for dispersal of these B cells throughout the tumor and better immune infiltration. And so this is just an example of the power of Bayesian data and how this can allow you to better understand cancer response and the tumor microenvironment and how the immune response goes into that. So our Bayesian gene expression platform really is a complete platform. We have a number of different assays that are contained within this platform, which we'll talk about today. So the first is our Bayesian fresh frozen assay. So this uses a polyadenylated capture method and is compatible with fresh frozen tissues. Next, we have our Bayesian for FFPE assay. So this is a probe-based method of capture, which allows us to work with FFPE samples, which tend to be more degraded than fresh frozen tissue. And these two versions of Bayesian fresh frozen and Bayesian from FFPE, we call these our direct placement assays. And what that means is that we have Bayesian slides, with these spatial barcodes. And for using these direct placement assays, you essentially section your tissue directly onto the Bayesian slide and capture your gene expression that way. As an alternative to that, we also have our Bayesian cytosys platform, which is our first Bayesian instrument, which we're very excited to talk about today. And so this allows you to actually use a different method compared to direct placement, and instead section your tissue onto a plain glass slide, and then transfer the analytes to the Bayesian slide using the Bayesian cytosys instrument. I will talk a little bit more about how that works in a few slides. Before I go into the different methods of capture, I want to talk about the Bayesian slide itself. So the Bayesian slide across all of our Bayesian platforms is very similar in nature. So we have the Bayesian slide with these capture areas. The capture areas will have different sizes. So we have the 6.5 by 6.5 millimeter capture areas. And with the Bayesian cytosys, we also have 11 by 11 capture areas as well. Within these capture areas, we have thousands of spatially barcoded spots. So each of these spots will have millions of oligos on the surface of that spot, and all of those oligos will have the same spatial barcode, which you can see here. And this allows us to tie all of the genes that we detect onto from that spot back to the spatial location on the Bayesian slide. So we put all of the data together via after next generation sequencing and analysis, we're able to see where the gene expression profiles originated from on the Bayesian slide. Our Bayesian spots are 55 microns in diameter, and so we estimate that this covers about 2 to 10 cells per spot. As I mentioned, we have different mechanisms of capture for the different assays. So we have a poly-A-based capture mechanism, which is compatible with fresh frozen tissue. Because this is poly-A-based, this is compatible with any species that have a poly-A tail, which is most of them. So you will be able to run a wide variety of species using the fresh frozen method. We also have probe-based capture, a probe-based capture method. And for our probe-based assays, we have human and mouse probe sets available, with the ability to design custom spike-in probes as well. The probe-based capture method works well with FFPE tissues, which tend to be more degraded, of course, but we've also shown compatibility with fresh frozen tissue as well as fixed frozen tissue recently using the Bayesian cytosys. Looking a bit further into the biochemistry of these different assays, so you can imagine the Bayesian slide, as you can see, the oligos on the Bayesian slide here. So they have this poly-DT sequence. So this allows for capture of polyadenylated transcripts from fresh frozen tissue. So that's pretty self-explanatory, I think, and we use reverse transcription to capture the mRNA fragments. On the other hand, we have our probe-based capture method. And the way that this works is we have essentially each of our probes is split into two. So we have a left-hand side of the probe and a right-hand side of the probe. And This is really nice because both sides of the probe actually need to hybridise to the mRNA target sequence in order for us to detect the presence of that sequence. So it really increases the specificity of the assay. So we've been using our probe-based capture methods. Both sides of the probe will hybridize to the mRNA transcript. We'll get ligation between the two. And then that results in this completed construct. You'll also observe that the right-hand side of the probe has this synthetic poly-A tail. And this allows us to capture that probe sequence on the Visium slide. So actually, it's the same slide architecture between the two assays, which I think is a nice feature. So it just makes it really easy to transition between one or the other. And today we're particularly excited to talk about our first Visium instrument, the Visium CytAssist. So we really see the Visium CytAssist as allowing us to bridge the worlds of histology and genomics, which traditionally can be kind of far apart and might not be the easiest to work together. So as I mentioned, for direct placement assays, you need to section the tissue directly onto the Visium slide. And for with amazing service providers like BioChain's, that's definitely they're very capable of doing that. But for more inexperienced users or people who maybe don't have as much experience in protomes or cross sections, microtomes or cross sections, that might be a bit more challenging. For you, I was kind of think of like, I don't know if anyone here on the game pilot wings when they were a kid, but you kind of have to jump out of a plane and just parachute and have to try to land in the landing area and not land outside the landing area. It's very doable if you have experience, but it can present some challenges. And so what's so great about the CytAssist is that you can just section your tissue onto a plain glass slide. So you don't have to do any sort of worrying about where exactly it gets. You don't have to land it perfectly on this little square. You can just section it onto a normal glass slide. Do imaging as normal, so your H&E or IR staining as normal. And then the analytes will actually transfer to the Visium slide inside the Spatial instrument. And so what this means for you guys is that if you have tissues of interest and you want to section them first and analyze them using H&E staining and determine which section is the section that you want to go further on and study using Visium, you can do this on your end and then actually ship just your sections on plain glass slides to BioChain for them to profile using the Visium assay. So this really streamlines the workflow and makes it easier for our customers and additionally easier for BioChain to work with samples. So you can send slides to BioChain or tissue blocks. I think whatever works will work with them. And we're happy to have further discussion about what makes the most sense for individuals' experiments. Yeah, and so we see the CytAssist as the way to get the most possible information from your samples in the easiest possible way. So the first way that this really expands access is through expanded sample formats. So as I mentioned, it's compatible with multiple different tissue types. So we've seen that the CytAssist, we've shown compatibility with FFPE tissue, fresh frozen tissue, as well as fixed frozen tissue. Whereas previously we only had compatibility with FFPE using our probe-based methods. With the CytAssist, we can access a lot more tissue types. Additionally, the quality of the tissue can be lower, so you don't have to have as high quality tissue using the CytAssist. We've seen that fresh frozen tissue with a Vim4 X4 is actually compatible with our CytAssist assays. Furthermore, again, you don't have to. You can use even archived tissue sections with the CytAssist. So you can take slides that have been archived for several months, and if you want to perform retrospective analysis, you can do that very easily using the CytAssist instrument. CytAssist is compatible with H&E and IF staining. So depending on which staining methods you are interested in or have experience with, those are compatible with downstream CytAssist analysis. We have expanded tissue size capture areas as well. So with our direct placement assays, our capture area sizes are 6.5 by 6.5. With CytAssist, you can now access larger capture areas. So we have an 11 by 11 capture area as well, which can allow you to capture something like the size of this, this entire area. And finally, this one I was the most excited about. So like I said, you can really choose the tissue section that you are most interested in prior to running the Visium assay. So again, if you think about it, you're using the rough placement, you kind of have to know in advance which section of tissue you're going to be interested in. And so you kind of have to know your tissue well enough to predict, okay, this section that's coming up is going to have the area of tumor that I really want to use. Using the cytosys, you can section tissues onto separate glass slides, perform H&E staining, and then use your pathologist or use your knowledge of the tissue to pick which of the several tissue sections that you just sliced off your tissue you want to analyze further. And so I think this is just a great way to enable users to make sure that they're going to get the most interesting and best possible data from those trends. The cytosys is designed to work seamlessly with existing histology workflows. So as I said, you would section your tissue of interest onto plain glass slides and then perform your downstream imaging again just on plain glass slides, the same as you would for any standard histological technique. The next step would be probe hybridization to your tissue. This step is again performed on a plain glass slide and using these cassettes that we provide. From there, once you perform probe hybridization, you would then load your tissue into the cytosys instrument and then analyze. So the probes will then transfer from your FFPE tissue down to the Visium slide, which has also been loaded into the instrument. And from there, you have probe extension and library construction, sequencing, and data analysis as normal. And so taking a closer look at what's actually happening inside the Visium, inside the cytosys instrument. So essentially, let me go back. So this section here of the instrument will actually flip up. And then you load your plain glass slides with the tissue sections on them into the top section of the instrument here. And what's really nice is you can actually just move the glass slides up and down essentially to fit within the capture area. And we have these blue lines and black lines here, which provide the guideposts so you can know exactly where to position your tissue sections so that it can align with the Visium slide. The Visium slide is then loaded into the bottom of the instrument here. And so what happens during the cytosys run is that we have the probes that label your 50 fixtures and your fixtures and tissue will transfer down during the cytosys run and be captured by the Visium slide on the bottom. And so that's how our cytosys instrument allows us to transfer data from a plain glass slide to a Visium slide in a seamless and easy to use manner. And so this enables a number of exciting things. I'm going to talk about a few use cases that I have here. So the first is we're able to look at retrospective analysis and analyze archived tissue slides. And so for this experiment, we wanted to see, can we get good quality data from sections of tissue that has just been sitting on slides for several months? So we took the section from a human lymph node, that had been FFPE processed, H&E stained, and archived at room temperature for about 5 months. And then we performed retrospective analysis on this tissue using our Visium cytosys instrument. And so here you can see the H&E stain of this tissue. So it's lymph nodes. This is a highly organized tissue type. So you can see within the lymph node tissue, you can see the presence of these germinal centers and things like that within the lymph node. And then we were really pleased to see that the cytosys data from this looks really beautiful. We were able to recapitulate these germinal centers, which have, you can see here in yellow and in purple as well. So using gene expression data, we're able to nicely call these germinal centers within the Visium data. We're also able to detect a variety of different cell types within the data. So dendritic cells, the T helper cells, as well as macrophage cells. And we were pleased to see that these are all really well expressed and quite spatially distinct within this tissue. So giving us confidence in the localization of these cell types. As I mentioned, you also have access to larger capture areas using the cytosys. So this is an example data set that we have from a human breast cancer punch biopsy. And so you can see that using the 6.5 by 6.5 millimeter capture area, we're able to nicely capture data from this punch biopsy. It's a highly heterogeneous tissue, and so we're able to observe that spatial localization and organization, as well as get that gene expression data. But using larger capture areas, we can capture even more of this tissue. And so depending on exactly what you're interested in looking at, or interested in doing something like, profiling of an entire mouse embryo, or you just have these really large, larger tumor sections that you want to examine are 11 by 11 capsid areas, maybe against it for your experiment. So these larger capsid areas allow for nearly 3 times the area to be captured. So, again, really nice for local tissue sizes. Finally, so I'm really excited about this one. So this is the recent release of our protein assay as well. So we know that a number of researchers really have this need for highly multiplex protein analysis in addition to RNA sequencing. And so we've been wanting to enable that for some time. We've just recently released our first protein panel. So we have a human immune cell protein panel available now. And so we've tested this on FFPE tissue, and we've seen really nice data, which I'll show in the next slide. But essentially, this capture is enabled through the use of these additional capture sequences on the Visium side, which are intended to capture barcoded antibodies. And so this is an example data set that we showed at AGBT, human tonsil. So here you can see this is also a highly organized tissue. So here you can see that we have the germinal centers within the tonsil here, which are nicely recapitulated using our gene expression clustering, and then also nicely recapitulated as well using the protein clustering in addition to that. And so our immune protein panel has 31 human immune cell markers, as well as 4 isotype controls. This includes cell surface protein and intracellular protein. We have this whole panel available, or information on the whole panel available at this QR code, but it's also something that we're happy to send you information on if you're interested in sending that project. And so I hope to show you today that there are many benefits to using the Visium CytoAssist instrument, including having your particular choice, your preferred choice of tissue. So you can really start with a number of different tissue types. You can start with tissue blocks or pre-section slides. Those are compatible with the CytoAssist. And it's compatible with FFPE, fresh frozen, fixed frozen tissues, and things like tissue microarrays. We've demonstrated compatibility across all those different sample types of CytoAssist. We want to give users a lot of confidence in the data that they get. And so CytoAssist instrument will allow you to profile small tissue sections with 6.5 and 6.5 capture areas, or larger tissue sections with our 11 by 11 size, as well as being able to capture gene expression and protein data so you can get the most possible information from your tissue sections. And finally, simplicity. So we really want to make Visium as easy to use for our customers. And so this allows you to manually pre-screen your tissues and easily ship to service providers such as BioChain to perform downstream gene expression analysis and integrate this workflow seamlessly into your existing histology workflows. The CytoAssist is really our feature of Visium. We're really excited about it. We've seen the kind of data that people have generated and are just really looking forward to what people are going to do next. But CytoAssist offers, again, improved assay performance, expanded sample compatibility, and co-detection of gene and protein expression. So really allowing for you to get the most possible from your respective samples. Okay, and so, naturally, you might wonder after doing this full transcriptome gene expression analysis of your tissue, what's next? What else can I do with my tissue? And so for that, we are launching our Xenium in situ instrument as well. And so our Xenium in situ assay allows us to take something like this very large FFPE cancer data set and turn this into a highly complex data set such as this, which you'll notice actually really resembles single cell data. And that's not an accident because essentially the data that you get is equivalent to single cell data. So our Xenium instrument allows us to better understand the relationship between cellular structure and function with an end-to-end in situ platform with an imaging-based readout. So one of the nice benefits about using an in situ readout rather than next generation sequencing readout is first, so cost reduction, so you don't need to pay for doing extra sequencing. The sequencing is essentially done on the tissue and it's a fluorescence-based readout. Additionally, time saving is also huge, I think. So you don't have to construct a library, send it out for sequencing, and then hope that your sequencing provider gets back to you or the time that they said they would. With the Xenium and C2 instrument, the data is going to be available immediately downloadable off the instrument. Our platform allows for profiling of hundreds of RNA targets with single cell or subcellular resolution. It's a very high throughput instrument, so our sample prep is very fast, just a couple of days of sample prep, and then just a few days on instrument to process the tissue. And additionally, our Xenium instrument is compatible with fresh frozen and FFPE tissue types. As I said, we've really tried to make this process as easy for users as possible. So you start by sectioning your tissue onto the Xenium slide. As I said, this can be fresh frozen or FFPE tissue. The next steps are permeabilization or deparaffinization of the tissue, depending on if you're doing fresh frozen or FFPE. And then probe hybridization. So you would do probe hybridization on the Xenium side, and then probe hybridization of the probe on the Xenium side. The next step would then be probe ligation. So we use padlock probes to again increase the specificity of the detection. So we ligate the two sides of the padlock probes together, and then we perform rolling circle amplification of that probe. And the nice thing about the rolling circle amplification is that this allows us to significantly amplify the signal to noise ratio. So we're better able to detect these probe signals using because we're able to amplify the signal out. So these steps are performed off instrument. The next, once that's completed, you would then load the Xenium slide into the Xenium instrument. And from there, the Xenium run will perform fluorescent probe hybridization and do a number of imaging and then decoding. So essentially, we're doing successive rounds of fluorescent probe hybridization, washing away and taking an image, and then doing decoding after that. And from there, you can immediately download that data from the Xenium instrument. And while our data types are very compatible, very open source, very compatible with third-party tools and analysis. But one of the things that I personally think is very cool about the Xenium system is how quickly you can actually get your hands on the data. So the decoding is performed actually in line during these imaging cycles. So you don't have to, like once the run is completed, you're able to access that data pretty much immediately because it's doing the decoding while the instrument is actually running. So you don't have to do any sort of like offline data analysis to get access to the data or anything like that. We provide ready-to-use standard file formats for further analysis. So again, it's going to be very compatible with community-based tools. It's, like I said, it is kind of similar to single cell data. You'll get this feature barcode matrix, which allows you to look at the gene expression profiles of the different cells within your sample very easily and be able to export those files to something like Syrah and do differential gene expression analysis very quickly. We also have our Xenium Explorer for an interactive visualization analysis, which you can perform off instrument. And I have a little video of the Xenium Explorer here. And so I really love the Xenium Explorer. It's really fun to use. It's super fun to sort of like dive into the data sets that we have. Xenium Explorer is available for free. And we have public data sets available if you want to check it out yourself. But in this little video here, you can see that essentially we have all the genes that we have in this panel, and we've divided them according to what cell types they are markers for. So within this cell type, we have myeloepithelial cells, stromal cells, and T cells. And we can just very easily click and say, okay, I want to see T cells. I want to see my stromal cells. And you can easily see which cells are localized where in the tissue. Furthermore, you can visualize the precise transcript localization and subcellular resolution of different transcripts. And so here in this video, you can see we're really able to visualize where this MS4A1 and BANK1 genes are. So those are our T cell markers. So we just very easily visualize where those are localized, even within a single cell. And the goal of all of this is to get you guys faster time to results. And this is an example of the type of data and analysis we would be able to achieve using Xenium. So here we have a very, very large breast cancer section. The section of the size of the section is about 10 millimeters by 20 millimeters, so that essentially covers the entire of our Xenium capture area. And there's approximately 1 million cells on this section. And so using our Xenium Explorer, you can easily dive deeper and deeper into the tissue until you're able to see something like this. So here we have these tumorigenic cells that are surrounding the tissue, and we're able to actually observe these cytotoxic T cells that have started to infiltrate the tumorigenic region of the tissue. And so this is the power of Xenium, is that you can really dive deep and see at a single cell level where your immune cells have infiltrated the tumor. And one of the standout benefits, I think, of using Summit technologies is this highly synergistic multi-platform analysis that you can do using our single-cell Visium and Xenium instrumentation. And so we have this really nice preprint available on bioRxiv. I do recommend checking it out because it has some really beautiful data. I'll just show a couple of data sets from that. But we were able to dig very deep into the structural carcinoma in situ breast cancer sample using chromium, Visium, and Xenium assays, and using this multifaceted approach, dive deeper into the tissue sample than really would have been possible using any of the technologies alone. So to perform this experiment, we took serial SFP sections and performed different assays using these serial sections. So we used our chromium single-cell gene expression flex assay, which is also a probe-based assay, which allows us to use SFP tissue sections as input for single-cell analysis. We also performed our Spatial assay using the cytosis. And finally, we performed our Xenium in situ assay on a serial section as well. And so this first data set is from our single-cell gene expression flex assay. And you can see that using our flex assay, we're able to identify all of these cell types within the tumor. So one of the things that we were able to identify was that there's not just one cancer signature within the tumor, right? So we have two different DCIS signatures, they're called DCIS1 and DCIS2. So this one is sort of in the more orangish color over here, and DCIS2 is over here in the pink. And we're also able to identify these invasive tumor cells in red. And so one of the things that I thought was really amazing to see was how well the Xenium data actually correlates beautifully to the single cell data. And so to me, if I was looking at this, so this data is actually from our Xenium run. I don't know off the top of my head how many cells we got from this data set. I think it was around 700,000. But you can really see how well this correlates with our single cell data, that just, is many magnitudes larger. So if you try to imagine how many rounds of single cell assay it would take to do 700,000 cells, if you do that just one Xenium run, it's really incredible. But what's even better about Xenium is that you're not just identifying these same cancer cells you can identify using single cell. We can now map them spatially to where they're originating from in the tumor. And so what you can see here is that, DCIS signature is localized more on the, or DCIS1 is localized more on the left-hand side, whereas DCIS2 is more on in the pink, is more on the right-hand side here. And then interestingly, you can see that this invasive tumor signature is more adjacent to the DCIS2 signature. And so this is a type of spatial analysis that you can do using the Xenium assay. So you can get both this really amazingly huge data set of like 1 million cells and be able to analyze it, again, using almost a single cell approach, but you can take that further and then also look at it in the spatial context as well. And that's the power of having these multiple data types available. And we are very committed to developing this platform further. We have a number of different innovations on our roadmap for Xenium. So I'll go through all of these. So first is next generation cell segmentation. So there are many different ways to think about doing cell segmentation using in situ profiling. We're looking at different stains to use and they'll be tissue specific. So we're working on different methods that will offer the customers for cell segmentation. We want to make this a multi-omic platform as well. So we are planning to add protein analytes to the Xenium platform in the next couple of months. Plexi increases is a huge one. So our goal is to increase it to, Jonathan, what's the goal for the Plexi for next year?
For next year, up to 5.
Up to 5,000, yeah. So our goal is to really significantly increase the complexity on the Xenium instrument. And then finally, I'm really excited about the students in metagnasal work. It's a lot of studying of different isoforms. And I know that this is often hard to answer the question. Studying isoforms, I think, is really challenging. But so our goal would be to have different programs available, or make it possible to study different isoforms within a given tissue, as well as even getting down to studying single nucleotide variants as well. So we're quite excited about those particular innovations on the roadmap as well. And then finally, I just want to end by saying thank you to everyone who has come here today to listen to us present on our technology, and thank you to everyone who has published or is thinking about publishing using our technology. Really, we make the tools, but you guys do the science and drive the innovation. We're just really humbled and proud of the number of publications that have been published using 10x technology. So we're currently at over 5,000 publications, and we're just super excited to see where you guys will next. So yeah, thank you.
So the next speech will be from our team, Dr. Rikita Dakkar, and she's an application scientist. She has worked. She earned her PhD in biotechnology, studying plant microbe interaction in saffron crops, unusual plant using transcriptomics approach. So the title of our presentation is going to be exploring the new frontier with spatial transcriptomics. Thank you.
Thank you. Thank you, Vidya. Hi, everyone. I hope you can all hear me. Can you hear me, Paul? Can you hear me? Okay. There you go. Thank you. Okay, so thank you, Vidya. Thank you, everyone. Thank you all for joining today. Thank you for coming out in person and over Zoom. My name is Rikita Dakkar, as Vidya told you all. And I joined the BioChain team about 7 months ago as an application scientist and in their sales and marketing team. And I'm very happy to have this opportunity to, walk you through the journey of our, how we started exploring spatial biology and where we are right now and where we expect to go. So we started this journey back in 2021 with 10x Spatial and Geomics platform from NanoString. And since then, we have grown along the way, overcoming the many road bumps that we came across and learning from them and expanding further to provide different spatial services all under one platform, all under one roof. So to begin with, I would first like to give you a brief introduction of what BioChain is, who we are. So BioChain has been engaged in providing biological samples since 1994. That's for the last 29 years. And we have been accelerating disease diagnosis and drug discovery research with complete solutions from sample collection to preparation and analysis. And our aim is to provide quality and innovative solutions. And we understand that no one solution fits all problems in science. So we are working along with you to custom design solutions to answer the specific questions that you have on hand. So the products and services offer under these 6 main categories. We offer biospecimens, which include frozen and FFPE tissues. These include sections, blocks, panels, and arrays, and nucleic acids and proteins. We also provide matched samples and custom procurement services as well. Our automated solutions include kits and instruments to make your day-to-day life easier, more specifically AnaPrep instrument and related kits. And we also offer reference and control standards, which include FDA arrays, nucleic acids, and proteins. Our most famous reference products are our Cyno monkey and Rhesus monkey products, besides others. And we offer OEM services for large life technology clients and also offer extraction and purification kits as well One of our hot selling product right now is cell-free DNA extraction kit, which has been proven for better yields amongst the competitors. So as I mentioned before, back in 2021, we took our first step in the spatial biology services and became certified service provider for the 10x Visium digital spatial gene expression service, as well as nanostring geomics digital spatial profiler. And we have since continued to expand our network of partners and collaborators in this field, and we hope to continue to do that. And so with that, for the sake of this presentation, I will expand further our experiences into the spatial biology services. And so as we began back in 2021, we quickly realized we can be an end-to-end service provider, with our in-house tissue repository, our pathologist, and scientific team. And we started with a few simple projects using our in-house tissue and TMAs to learn the steps that were involved in these workflows, the sample preparation and optimization and the entire workflow for both 10x Visium as well as nanostring geomics platform, all the way up to reporting and understanding the data. So before I head into some case studies, I want to briefly talk about our tissue procurement. And we have a repository of retrospective collections, which includes frozen and FFPE tissue. These are IRB approved and donor consented samples selected by our board certified pathologists. We provide match sets with tumor and normal adjacent tissue from the same donor and cancer cell lines and the clinical data associated with them. Also, we provide primary and metastasis samples, plus some rare disease tissues such as Alzheimer's, Parkinson's, glioblastoma. And besides our repository and prospective collections, we also work with the customer provided material. And, as an example, I'm sorry, give me a second here. I just want to make sure I cover everything and don't derail from the topic. It's just so easy to do that. Okay. So our first few experiments, we used some lung, brain, breast, and colorectal cancer and normal tissues from our repository for 10x Visium study and a breast cancer TMA for our geomic study. I will talk about the results from these studies in a few slides, I promise. But first, let's take a look at sample preparation and optimization, which is the next step once you have your tissue. So let's first talk about sample preparation for 10x Visium. Frozen tissue, 10x Visium sample preparation can look very different depending on the type of tissue. So like frozen tissue with direct placement requires OCT embedding and tissue optimization. Tissue optimization, as Samantha just was explaining, it can be difficult even for histopathologist, experienced histopathologist. Tissue optimization and slide preparation is a very tedious and time-consuming process. So in the beginning, it seems like FFPE, fresh cut was a more quicker option as far as the workflow was concerned. Also, with direct placement, rinse score or DV200 qualification, which is much higher, 7 for rinse score and 50% for DV200. This was a limitation for some of our customers who were working with some rare and difficult tissues. So fortunately, these issues were addressed with CytAssist, and as Vidya mentioned, we took a leap and bought the CytAssist, and which does not require tissue optimization for frozen samples and can take rinse score as low as 4 or DV200 as low as 30%. In addition, we can also use archival FFPE tissues and TMAs with CytAssist. So earlier this year, we added CytAssist to our services, which also has apparent benefit of high coverage of the capture area compared to the direct placement, and much higher sensitivity too. So as a case study with CytAssist, we were able to resolve some of the issues that our customers were facing. For example, one of our projects was delayed by 7 months as the customer wasn't able to find a good rinse for glioblastoma tissues. They were working on fresh glioblastoma, surgery tissues, and it was really hard to get a good rinse for sample. And ultimately, we decided to move forward with the project using CytAssist and use the precious samples that they had obtained with low rinse score. Another one of our customers working with diseased and healthy arteries and aged patients had similar rinse challenges besides low tissue amount. So using CytAssist, we avoid tissue optimization, which needs more tissue slides as opposed to just one slide required by CytAssist, and help this customer. In yet another example, we were able to use archival slides from our customer to generate results using CytAssist. So it has really proven very beneficial to add CytAssist to our repertoire. And moving on, as I promised to show the results of our preliminary studies, here is an example of our lung sample characterized using 10x Squeezium technology. The image here shows the tissue in the capture area and UMI plots and clustering. I would like to further talk about a poster that we recently presented at AACR in April this year. And we used our in-house colorectal cancer and normal samples for this study to provide insights into tumor microenvironment, vis-a-vis heterogeneity and spatial organization of cells. For the sake of time, I am only highlighting a few key results here, but please feel free to follow up with our technical team later today or via email if you are interested in learning more. The figure here shows the heat map, which clearly shows the differential gene expression, the differential upregulation in the tumor below, in the second one. The below tissue is the tumor and the upper one is the normal. And you can clearly see differential genes upregulated in tumor versus the normal sample. So these violin plots further show upregulation of genes in comparison to the normal tissue. And these upregulated genes also consisted of these 5 well-known CRC biomarkers. Further, this violin plot here shows the upregulation of B-cell associated immune cell biomarkers. And again, we can see we found out that some of the very well-known biomarkers were upregulated in the tumor tissue. So coming back to sample preparation and optimization, let's talk about geomics now. So with geomics workflow, building a microarray with multiple treatment or tumor samples is cost efficient in terms of reagent consumption. So we can spatially profile multiple tissues on one slide as opposed to multiple runs. And we do offer TMA constructions as shown in these pictures above. And so we can spatially profile multiple tissues on one slide. And as an example, our in-house pathologist recently helped build a TMA with 20 cores. And what they did was they wanted to study breast tissue. And within the same tissue, they wanted to study progression within the normal areas compared to the tumor areas. So our pathologist helped select the cores and build a TMA. Another example is that of our in-house TMA that we use to characterize using geomics platform. And I will talk about it in just a few minutes. But before that, I would like to talk about morphology markers, selection and optimization, as well as proteinase K concentration and optimization, which is one of the key steps in geomics workflow. So morphology markers are key in the genomics workflow, as they are used to select ROI and guide further tissue segmentation, as they're specific to the cells of interest in the tissue under examination. And with each different sample or treatment, the concentration of morphology markers and proteinase K needs to be optimized to enable clear visualization of the tissue and segmentation within the tissue. So depending on the tissue, this may take as less as 1 dry run or more. And here is the breast tumor TMA that we characterized using genomics. And this TMA is, we built this TMA at BioChain, and this comprises of 25, these from the second row to the last, 25 breast tumor tissues in duplicate, plus 4 normal breast tissues in duplicate, and 1 placenta tissue for negative control. We used the morphology markers PAN-CK to stain epithelial cells green, and CD45 to stain immune cells red. So for ROI selection, in the whole breast TMA, we selected 88 ROI, that is regions of interest, and each being 400 microns. In the image here, I'm showing you 2 ROIs in particular, B1 on the top and C5 on the bottom. So the left image here shows, the epithelial cells stained green with PAN-CK, and the red area that you see is immune cells stained with CD45. And on the image on the right here, you see blue and yellow colors. Those are the segments that we selected. Blue segment indicates the segment that has only the epithelial cells, which is PAN-CK positive, CD45 negative, while the yellow indicates only the immune cells. So further, we used the data generated from here to create these volcano plots and violin plots. And clearly here in the plot on the left, you can see the differential gene expression between the immune segment versus the epithelial segment. And then the violin plot here is showing the gene expression for only ERBB2 gene in particular. This is the gene that encodes HER2 protein in breast cancer. And, you can see here that this particular gene was upregulated in the epithelial segment in the tumor tissue, tumor cores, compared to normal. So when it comes to spatial workflow, we further have added a few more technologies. We started with NanoString Geomics DSP and 10x VZM direct placement. Then we added Site Assist and SlideSeek from Curia Bio. And then we added Codex in partnership with Enable Medicine and StereoSeek as well. To summarize, I want to go over some case studies and future prospects. We have successfully been able to process precious customer samples and, generated the data and completed projects with both Geomics and NanoString platforms. And we have been able to construct TMAs, help the customers with histopathology, and use our in-house FFPE tissues and TMAs to pre-characterize these tissues. And these pre-characterized tissues are also available online for purchase with the section and the data. And we have successfully used archived hardcover slipped slides as well with 10x Site Assist. And also more recently, we are now getting into further characterizing our pre-characterized 10x pre-characterized oncology samples with analysis such as codec with the help of our Enable team. So that's something I know Gulpreet mentioned a similar project too. So that's something we are heading into now. And in the coming few months, we are looking at doing more of such projects where we are combining different technologies and, yielding more, generating more results. And we are very excited to further onboard proteomics and StereoSeq technology as well. And to conclude, I just want to highlight a few of our key strengths, which are our biorepository, which comprises of IRB approved rare tissues as well, our flexibility to work with different kinds of tissues, and our experience in tissue handling, TMA construction, and our sample preparation, and our in-house board certified pathologists, scientists, and the pre-characterized sections that you can purchase online, as well as all these multiple platforms being in one place. So with that, I would really like to thank all of you for your patient listening. If you have any questions, please feel free to ask and reach out our technical team as well. And thank you so much.
STOMICS, this is the new kid on the block, okay, and which is basically the other name. I didn't know STOMICS, and somebody said StereoSeq, and I was able to get both of these. Okay, it made sense to me. So John Manuel, who is the director of business development for Spatial Genomics at Complete Genomics, based out of San Jose, will be giving us the talk. So he has worked for a long time at Fails, and also for 20 years experience in anatomical surgical pathology. So John graduated from University of Washington. So I welcome John to talk about StereoSeq.
You want to have two laptops because I didn't transfer my notes properly. Happens sometimes. All right, so if I am not loud enough, please raise your hand. I'll be happy to speak up. Just let me know. And yeah, thank you for being here. As Vijay mentioned, I'm the director of BD for the STOMICS platform. First, I want to thank you all for being here, for your interest in spatial biology. And, there are a lot of good solutions here. And I think it's, one thing we're all aligned is that we're excited to move you forward with your experiments. And I do want to thank Vidya and BioChain for getting us in. We were a little late to the party today, but we were able to get in. So we're excited for that as well. I've had the privilege to work with BioChain for a couple of years now. Really strong group, technical, very cracked team with their technical expertise, as you already know now. They have a pathologist on site, and they do TMAs, which is tough to find actually right now. So that's good to know that you have that option. All right, so let's dig in here. So what I want to look at first, or just touch on, is a little bit of the outline. First thing I'll do is talk over some applications of how StereoSeq has been used to date. A high-level look at the technology. We actually have some technical experts here. We have our innovation technology manager, Dr. David Taussig, he's down here. And then we also have Dr. Jiang, who's our VP of research. She's in the back in the corner right now. And we'd be happy to take some deeper technical questions once we wrap up today. I'll talk about what we have in store for the future, and also some of, who your local team is, and, how we work with BioChain. We're very excited to have them as our first CRO partner. All right, so what you're looking at here, this is a cover from Cell from 2022, about spring of last year, and this shows data from a mouse organogenesis study. So the researchers here, what they did was they utilized StereoSeq technology to enable a mouse organogenesis atlas based on whole transcriptome data at single cell scale. So that'll be a little bit of a motif, a little bit of a theme I'm going to peg on here, is that whole transcriptome in single cell. That's a unique aspect about this technology. This was accomplished with the use of DNA nanoball pattern array, which you see on the top left here. It's a very high level look at the workflow, and I'll go through that in some greater detail in a little bit. We combine that with RNA capture from overlying tissue, permeabilize that tissue, capture that RNA, create a library, and submit that for sequencing. Once you have your sequencing readout, that's overlaid with an image that's taken upstream. Now, the data you're seeing here, what the researchers were able to do with the brain data was identify over 50,000 segmented cells utilizing cell segmentation software, average UMI per cell of 1,900 and 792 genes per cell. With the embryonic tissue, what they found was in segmented cells, over 280,000 segmented cells, average UMI of 1,100 and 529 genes per cell. 48 tissue types are identified through the developmental process. You see over here, a few days, 53 sections were looked at. And this atlas now, this will help enable in-depth investigation of long-standing questions related to mammalian development, whether it's abnormal or normal development. All right, so last fall, a little more recently, we were covered in Science, and this was for the first spatiotemporal atlas of the axolotl amphibian brain. Excuse me for a second here. Yeah, so what the researchers did here, they utilized stereo-seq technology to spatially resolve single-cell transcriptome for the adult axolotl telencephalon. Users generated spatial transcriptomic data across multiple sections that were taken from this specimen. And they looked at areas that were where they induced injury, and looked at developmental stages and also regenerative stages to the site where they introduced that injury. The data, based on the single cell resolution that's offered with this technology, allowed the researchers to identify 33 cell types during the developmental process, and specifically identified 28 cell types involved in the regeneration process where they had introduced injury. And this includes types of excitatory neurons, inhibitory neurons, also several ependymal glial cell types also. Now, this is the latest. This was actually just recently put out on Nature. This is a Cell Research publication just 2 days ago. This was put out by Nature. This is a study where researchers characterize the spatial transcriptional landscape of ICC. So intrahepatic cholangiocarcinoma specimens, those are surgical specimens, clinical specimens looking at solid tumor. The samples included areas of solid tumor, adjacent unremarkable liver tissue, and the margin as well. So margin is a big interest right now for specimens like this, especially in terms of what's happening at that gene expression level. In as little as a 500 micron wide section, which was centered bilaterally on the tumor and tissue boundary where you have that invasion, they were able to identify enrichment of distinct immune cells, suppressive immune microenvironment, and metabolic reprogramming of tumor cells. They also identified different stages of damage in hepatocytes with expression of SAA, that's serum amyloid A, which is known to activate macrophages and plays a role in invasion. So that's something that they can look at more closely as well. And anytime you have that invasive process at these tumor fronts, that leads to a worse prognosis. So that's something they can focus on. But what's unique here is it's not just that spatial information, that gene expression data, it is combining that single cell location, identifying where those single cells are located alongside that whole transcriptome information. So this type of information is impactful, of course, for biological insights for specimens like this, solid tumor specimens, and opens up the options for developing novel therapeutic strategies for specimens like this, particularly solid tumor specimens. Why stomics, right? What makes it different? One, it is unbiased whole transcriptome. Any section that's placed on the chip will be analyzed. It will be permeabilized, and then the RNA is captured from that entire section. Another unique aspect is the size of that spot. We're at nanoscale resolution. Each spot is 220 nanometers in diameter. So a 1 by 1 centimeter area or chip that we have, that's our smallest chip, is 1 by 1 centimeter, contains 400 million spots. So about a 10 micron cell will cover about 300 to 400 spots, depending on the shape, a little bit of the boundary of it. Here's an example. Tissue has been stained, the nucleus has been stained, and every spot that you see on that nucleus is one of the capture spots. So to give you an idea, this is about a couple hundred spots under one nuclei here. Nucleus. Additionally, we have single cell resolution. This is something where we do separate ourselves and that we have software to establish boundaries on our cells. We have cell segmentation software. It's nucleus-based expansion method, so we can put a boundary and explore the tissue and look at those specific cells, seeing where they're involved in regeneration, tissue injury, where they're involved maybe recruitment of macrophages or other malignant processes. And then unmatched field of view. This is a unique aspect for our technology too. As I mentioned, our pre-designed chip, the smallest one is 1 by 1 centimeter. We also have a pre-designed one that's 2 by 3 centimeters. However, we can go larger. As an example, I believe this is one hemisphere of a macaque brain that was sectioned and placed on one of our chips. It's about 7 by 7 centimeters, but we can go all the way up to 13.2 by 13.2 centimeters square. So this can accommodate multiple smaller sections or one large section of tissue. In terms of analysis, what's really interesting is we can get down to one spot to analyze that spot with our software. As I mentioned, 220 nanometers is the diameter. Center to center distance is 500 nanometers. What you're seeing on the right is a recognition of the bins, how we assign these. One bin is one spot, 2 bin is 2 by 2 spots, and so forth. Not until you get up to bin 20, where you're starting to approximate what we know is roughly a single cell size. So you have about 400 spots for a single cell of that size. So that's pretty remarkable for the type of data you can pull out just from one cell based on the number of spots covered. Bin 50 is up to 25 microns. If you want to go that large for some reason, or if you want to approximate something that's 100 microns, you can also analyze that tissue in that way too. Here's what the chip looks like. Each slide has its own chip. We used to have 2 on there, but we brought it down to 1. It's easier to work with. And the tissue is placed onto that chip. The chip, as I mentioned before, each has 400 million spots to it, 220 nanometers in diameter, and the spacing of 500 nanometers center to center. And each has a DNA nanoball on there. So these are pre-designed chips. As they come out, they have that nanoball technology on there. And from that, you have probes. There are about 200 probes for each spot, and that equates to trillions of probes for a 1 by 1 centimeter chip. And here's a general look at the probe design. It is a poly-A capture of RNA from the tissue on top that we permeabilize. And I'll show you a quick look at that workflow here. So the way this works is that tissue is sectioned directly onto the chip. Once it's onto the chip, we permeabilize that tissue. Right now, this is FFPE, fresh frozen friendly, but we have FFPE coming out later this year. So that's something to look forward to, and we can talk more about that towards the end of this presentation. Once it's permeabilized, RNA diffuses down to the surface, and that's where we have that poly-A capture. Downstream of that, there's library generation. Submit that library for sequencing. And once you have your sequencing readout, that is overlaid with that nuclei stain up front. So you're able to segment those cells and then overlay your transcriptional data. So some of these tissues that we have sampled are here. And right now I say some because this is well beyond this at this point for human tissues, mouse tissues, and other species, which I'll talk about. But for human, as you can see, some of these tend to be challenging specimens. You have lymph node in there, fatty liver, NASH liver you may be working with, metastatic lymph node, brain cancer. We've had very good success with these type of specimens, and we have also worked with a variety of mouse tissues as well. We're not limited to there, of course. With fresh frozen direct poly-A capture, we can work with a variety of plants, and we have great publications and data on insects, on fish, and definitely on macaque tissue as well. In terms of what's coming up in the future, we will have some new protocols. As I mentioned, FFPE is in the future. That will be towards the end of this year. This will be compatible with tissues that have been fixed in PFA, 4% PFA, or 10% MDF. And from that point on, embedded in paraffin. Higher single cell resolution, we're always working on our slide chemistry to improve it. We're very happy with where we are right now. We have really good data, but we're always looking to improve. In terms of imputation with H&E and IF, this is something currently we use an adjacent section, but next month we will be offering H&E and IF on the same tissue section. So right now the adjacent section works very well, but we know true multi-omics on the same section, that's ideal, and that's the direction we're moving to. With the multi-protein profiling, this will be coming out, I believe, towards the end of this year, and don't hold me to that. There's some more details to flesh out there, but that will be a 20+ multi-protein profiling panel. And then algorithms and tools, we're always working on our software, improving what we have in that regard. Cell binning analysis, we're going to bring some efficiency to, and also 3D reconstruction. That's something we can do right now. There was a pretty big publication relatively recently, or preprint, on macaque brain, where they looked at several hundred sections of macaque brain with 500 micron spacing between them and did a 3D reconstruction. So you had a transcriptomically profiled region of brain in a 3D, in a way you could appreciate in a three-dimensional way. Here's a quick look at what we have for a multiplex IF, some proof of data concept we have. We've marked CD45R and CD4 here, along with DAPI. DAPI will be one of the things that's required. And these can be merged. As you see here, we get really nice data. We have our spatial transcriptomic data, and you can have this IF image also. What's unique about this is you can raise the number of stains you want. You can go up to 5 and then still include DAPI, so have 6 colors that you're working with in total. And then with H&E, as I mentioned, we have H&E coming out also next month. So this will allow you to appreciate that morphology on the exact same tissue section that you're working with and completing your spatial transcriptomics, generating the library. So that is what I have, just to give you a quick intro onto Peak and, what still makes what we offer. We're working with BioChain, as I mentioned, pretty closely. We have some good data already on some samples that we've worked on, and we'll have a more comprehensive presentation coming out probably in like 6 to 8 weeks. We'll be planning something, but I do want to thank you for your presence here and for your interest, and we'll be happy to take some questions. I may recruit some help from our technical manager and VP for research as well, but yeah, happy to take any questions you can have.
I really appreciate all the people who joined in via Zoom, as well as people who drove in. And, we really would appreciate if you have any questions, come talk to us, any projects, anything, just come talk to our team is here. The NanoStream team is here, Atomics team, the 10X Museums team, and the Enable team.